How do you study a medical mystery? Sometimes we can see the symptoms of a disease, but finding its cause is like navigating a maze. Scientists must investigate from different angles, adapting their approach when they encounter a roadblock.
Then, of course, what happens is you run into a problem, and things were going great, and then we tried to do something else, and that didn't work. We stepped back again, and next thing went really, really great, and then we tried to do something else, and that didn't work.
One disease that poses this challenge is Parkinson's, a disorder that destroys brain cells and the ability to control movements.
Well, it's a very slowly progressing disease. It occurs in the nervous system, which is inherently a very difficult place to study. By the time someone presents with the disease, the particular cell type that is most affected by the disease, about 80% of them are already gone.
That's one dead end. Since they can't study the brain cells of patients, the Lindquist lab turned to an unlikely source: yeast.
It turns out to be that particular organism is the best studied organism on Earth. You can do amazing things with it, and you can do them fast.
Postdoc Josh Kritzer wants to give yeast Parkinson's disease. That's not easy since researchers still don't know what causes the disease, but they do have a prime suspect.
When you look at these neurons that are specifically affected by Parkinson's disease, you see within them these piles of debris. And what we find is that they're made up very substantially of this one protein, alpha-synuclein. And so we say, aha, we know that alpha-synuclein was involved in some sort of horrible accident in these cells. But that doesn't necessarily tell us whether alpha-synuclein was the instigator of that accident, whether it was a bystander or enabler of that accident.
Betting that alpha-synuclein is the primary cause of Parkinson's, Josh loaded up yeast cells with the suspect. It killed them. His challenge was then to find a drug that would rescue the yeast.
The traditional approach to drug discovery is to basically have a robot churn through as many drug-like compounds as possible. This is very expensive and time-consuming.
Instead, Josh got the yeast to do the work for him.
I settled on a novel method of tricking the cells in making those compounds in the first place so that we don't have to sit here as chemists and synthesize every single molecule.
Josh made his yeast cells snip out tiny bits of their own proteins called cyclic peptides. Each cyclic peptide is a potential homegrown drug.
So in an initial screen of over 5 million cyclic peptides, I've managed to find 2 cyclic peptides out of those 5 million that specifically suppress the toxicity of alpha-synuclein in yeast.
Those 2 peptides effectively cured yeast of Parkinson's disease. But that's a long way from a human therapy. That's where Chris Pacheco comes in. His goal is to test Josh's cyclic peptides in human cells. But before he can get there, he has to see if alpha-synuclein will even make the human cells sick, like it did in yeast.
People have been trying to overexpress it in human cells and trying to kill human cells with it, but it's been a challenge, it's been a hurdle. I just couldn't believe it. I just couldn't believe that if you overexpressed this protein that is toxic to yeast and toxic to humans, I just couldn't believe that it was not going to be toxic to these cells.
For months, he set up trial after trial to make his cells sick.
You don't understand, the thing is, it takes so long to do this, right? And then it's always just little, you see a little bump here and you're like, ooh, maybe that's it. And then you try to repeat it and it doesn't repeat.
A few weeks ago, his cells finally got sick. That's a long-awaited step towards simulating Parkinson's, but it's still early and much of the mystery remains. Can Josh's peptides save Chris's sick human cells? Will their efforts lead to a human cure?
I have to feel that it'll be done in our lifetimes, that we're figuring this thing out, and that we're just learning the proper approaches to answer these questions.